done, which is of 30 seconds to 1 minute. Next wash. After that we are adding a alcohol here, and this alcohol is mentioned as decolorization agent. Okay, decolorization agent, and which is of 20 to 25%. We can use sulfuric acid or acid alcohol also. And the next is counter stain, which is methylene blue or malachite green, which is of 1 to 2 minutes of duration. And after that we have to air dry this slide. And the very next process is we have to perform the microscopic observation. So in this microscopic observation, we are going to learn like what kind of bacteria it is based on the size, shape and color. So based upon the size, shape and color. Shape, size and color, right? So based upon that we are going to find out what kind of organism it is. So here we are going to identify a pink color. So pink or red. It is in rod shape. And here it is acid fast. So the pink color rod shaped bacteria indicates acid fast. Okay. So in the next diagram we can identify clearly, that is the rods are appearing in here, it is in pink color, and the remaining part we can, so here it is visible in blue color. Why? Because due to the counter stain. Why? Methylene also we have added at the end. So the primary stain is going to intact, or it is going to retain or resist with the with the cell wall of mycobacterium. Why? It doesn't intake any kind of methylene blue. So the outer membrane or the outer part of this smear visibility shows in methylene blue color, and the main part, which is to identify the bacteria, it is shown in a rod shape, which is in pink color. So by this we are going to identify the bacteria. Okay, so I think we have understood that acid fast and non-acid fast difference. So pink rod shape, it indicates acid fast, which is of Mycobacterium tuberculosis. Okay, so in the very first beginning in introduction I have said you that it is introduced by Ehrlich and it is modified by Gill and Nielsen. So what is the modification we have I have told before itself. So now we are going to see the modification. That is nothing but in tuberculosis, in Mycobacterium tuberculosis, we are using a decolorization agent, that is alcohol, which is of sulfuric acid H2SO4 decolorization agent of 20 to 25%. Okay, and the Mycobacterium tuberculosis and lepra bacilli is also having the same mycolic acid, which is in high waxy content, fatty acid and lipid also. But the only differentiation is the lepra bacilli, the cell wall, which can easily decolorize, and it's only required 5% of sulfuric acid. So this is the thing which Gill and Nielsen has modified in acid fast staining. Not only for lepra bacilli, and for many kinds of bacteria it is based on 1 or 2% or 3%. It varies, depends upon the time duration and the cell wall content in that. So this is the thing we have discussed, and the next I am going to show some examples of acid fast organisms. Like for bacteria, Mycobacterium, Nocardia, Rhodococcus, Sigma bacilli and the last, and the other is also bacterial spores, spermatic head. In parasite, oocyst of cyclospora and isospora. So these are some of the organisms which undergoes in acid fast staining technique. Okay, so this is the topic of acid fast staining technique. Thank you.